Okay, great. Can everybody hear me?
Yep.
Okay, perfect. Thank you. So today I will be talking about something slightly different, something that's been on the mind of Alzheimer's community for a pretty long time. It's a really long story, but I think finally it's winding down. So the title of my talk today is A Cure in Mind, A Cure in Sight, the aducanumab story. And I will discuss a couple of studies today that resulted from collaboration with Biogen, the pharmaceutical company that is currently developing aducanumab as a disease-modifying therapy for Alzheimer's disease. So I would like to dedicate this talk to Alzheimer's patients and their families because they have been extremely patient waiting for a cure. As you know, there is no cure for Alzheimer's disease. There's some symptomatic treatments, however, there is no disease-modifying therapy. And I hope I will convince you today that we are very close to that point. So a brief introduction of Alzheimer's disease. As a lot of you know, Alzheimer's disease is a progressive neurodegenerative disorder characterized by deposition of amyloid plaques in the brain parenchyma and intracellular hyperphosphorylated neurofibrillary tangles. As Chris already mentioned, there's substantial cell death as part of the disease process. But in addition to these major hallmarks of Alzheimer's disease, there's been subtle changes characterized in the laboratory, specifically of my postdoctoral advisor, Brian Baskey, and that's something that I've adopted in my laboratory. We see disruptions in neuronal calcium homeostasis. That is, as you know, neurons, they maintain the intracellular calcium levels within a very tight range for normal function. And in Alzheimer's disease in particular, there is disruption in calcium homeostasis, which results in abnormally elevated levels of neuronal calcium, what we call calcium overload. So we're able to monitor this calcium overload and use therapeutic approaches to try to reverse this calcium overload, thus getting a readout of treatment efficacy. So anti-amyloid beta immunotherapy research efforts have been around for a very long time and initially showed great promise, especially in animal models of Alzheimer's disease. These are just a few papers that I'm mentioning here, but there's been a great number of publications showing that this is a promising venue to pursue. However, some of the immunotherapy trials, including solanezumab and bempanezumab, are just some that I'm mentioning here, have failed phase 3 clinical trials. So there's been this challenge to translate the success in the mouse models into humans. So Biogen started developing aducanumab, which is an anti-beta monoclonal antibody, which is slightly different from other antibodies. The reason is it is a fully human antibody. So it was originally isolated from centennials, these individuals that have long lifespans and are protected against disorders, including Alzheimer's disease. So Biogen has adopted this technology through reverse translational medicine technology to not only isolate the antibody, screen it against human tissue containing amyloid plaques, develop this antibody through recombinant production, treat it in vitro as well as in vivo animal models, and they started using it in clinical trials with patients. So when the results of phase 1b clinical trial first came out, it generated a lot of excitement in the field. And my postdoctoral advisor, our very own Brian Baskey at Massachusetts General Hospital and Harvard Medical School, when asked for an interview for Fortune magazine, he said, "This is the most successful clinical trial in Alzheimer's disease ever." And the reason behind his enthusiasm was that in addition to decrease in amyloid, as shown here, this is an image of Alzheimer's patient before aducanumab treatment, and this is after treatment with the antibody for 52 weeks. They saw a decrease in amyloid on the PET scan. In addition to decrease in amyloid, they also saw a dose-dependent decrease in cognitive decline. So the cognitive decline in these Alzheimer's patients was slowed as a result of immunotherapy treatment. And this was the first case where this was reported. This was the first antibody that achieved this. So today I will discuss two studies that resulted from collaboration with Biogen. The aim of the first study was to determine if treatment with aducanumab, anti-amyloid beta antibody, leads to plaque clearance and restoration of neurite calcium homeostasis in TG2576 mice. And here we used multiphoton microscopy to look at the amyloid inside these mouse brains before and after antibody treatment. And we also performed calcium imaging using a ratiometric calcium sensor that I will introduce, yellow chameleon 3.6, that allows us to distinguish and determine the absolute baseline calcium levels within neurons. So you can see the healthy neurons are shown in blue, and neurons that have calcium elevations or calcium overload are shown in red. The aim of the second study was to determine the aducanumab mechanism of action, and that work is still currently ongoing. There we turned to neuronal cell cultures where we expressed the calcium sensors there to see whether aducanumab has protective roles. So onto the first study. We used very old, 22-month-old TG2576 mice that we injected with methoxyXL4, which crossed the blood-brain barrier and bound to amyloid plaques, which we could then visualize through cranial windows. To achieve a cranial window, we performed craniotomy, durotomy, and topically applied aducanumab directly to the mouse brain. And then we sealed the cortex with a glass cover slip, thus making a cranial window. Right after the surgery, we performed multiphoton microscopy imaging. So imaging session 1 would be the baseline imaging session. And then we allowed these mice to recover for 3 weeks. We sent them back to their cages. And 3 weeks later, we performed a second imaging session to determine the effect of antibody treatment. And we imaged amyloid plaques with multiphoton microscopy. And the working hypothesis was that an acute treatment with aducanumab would trigger amyloid plaque clearance. So in my laboratory, we use multiphoton microscopy. And just to give you an idea, this is a window into a mouse brain, TG2576 mouse brain. And you can see there are these amyloid plaques that are present. And there's already some deposition of amyloid along the blood vessels, what we call CAA, cerebral amyloid angiopathy, shown here. So when we applied aducanumab to the brain, we saw beautiful colocalization of the antibody shown in red with the plaques, which you just saw in green, making yellow. So antibody does engage the target. It does localize to amyloid plaques in addition to CAA, as can be seen here. Okay, so here is the results of our studies. We, as I mentioned, we monitored the same animals before antibody treatment and after antibody treatment. And the reason for that is we wanted to find the same amyloid plaques and what happened to those in the same animals. So here we're looking at these two amyloid plaques that were cleared as a result of aducanumab treatment. So antibody was effective at clearing plaques. However, it did not prevent deposition of new plaques. As you can see here, there's no plaque present. And then 3 weeks later, we saw amyloid plaques. Our control antibody that we used got into the brain. However, it didn't have a known epitope in a mouse brain. So that was an antibody treatment with the control antibody, and that treatment did not result in plaque clearance. As you can see, there's a cluster of amyloid plaques that are present after control antibody treatment. So the effect on plaque clearance that we saw was selective for aducanumab. There was a statistically significant increase in plaque disappearance as part of aducanumab treatment compared to the control. However, there was no difference, no statistical difference in new plaque appearance. Therefore, aducanumab substantially decreased amyloid plaque burden, while the control antibody led to increase in amyloid. So then we set out to carry out a chronic treatment where we treated our animals for the duration of 6 months, shown here. We would inject the antibody, either aducanumab or control, intraperitoneally at 10 mg/kg weekly. And prior to the treatment, we took 17 to 18-month-old animals, and we expressed yellow chameleon through viral delivery, performed craniotomy, allowed the virus to express for 3 weeks, and then we would inject methoxy and image amyloid as well as yellow chameleon. And then right after that first imaging session, we would start antibody treatment. And then we would re-image these animals regularly until the end of the treatment. And there we visualized calcium reporter yellow chameleon 3.6 with multiphoton microscopy. And the working hypothesis was that chronic treatment with aducanumab would lead to a normalization of calcium homeostasis. So for those of you that are not familiar with yellow chameleon, let me introduce you to yellow chameleon. It is a FRET-based probe. It has a CFP and YFP connected by a linker. And the ratio of YFP to CFP is actually able to tell us the absolute calcium concentration within the neurons. The higher is the ratio, the greater is the calcium concentration. Previous research in the lab has shown that it is the amyloid beta that is responsible for intracellular calcium elevations. As you can see here is an example of APPPS1 mouse that we used, and we saw these sick neurons that have calcium elevations shown in orange and in red. And we also verified that it was the oligomeric amyloid beta that led to calcium elevations. Here's an example of a healthy wild-type mouse that was treated with A beta oligomers that resulted in upregulation of neuronal calcium. And these are just the histograms. This is how we represent the data in the wild-type animals. There are very few neurons that have elevated levels of calcium, while in APPPS1 animals, there's close to 20% of neurons exhibiting calcium elevations. So we verified that in our hands, we saw calcium overload in TG2576 mice. Again, these are high ratio of yellow chameleon and hence high calcium concentrations within neurons. So prior to immunotherapy, the neurons of our animals exhibited calcium overload consistent with previous work. So what happened as a result of antibody treatment? Here's is an example of a neuron that started out with high levels of calcium, and as a result of aducanumab treatment, we saw restoration of baseline calcium to normal. And as you can see here, the blue neurons have calcium concentration around 100 nanomolar, which is considered normal. In our control antibody treated animals, again, these are the animals that were treated with antibody that did not engage amyloid plaques. And what we found was that there are these neurons with calcium overload, and those remained overloaded in the course of the treatment. So neuronal calcium could and was used as a functional outcome measure of treatment efficacy that we saw with aducanumab. And actually this restoration of neuronal calcium was quite robust. Within 2 weeks, we saw a substantial drop in the percentage of neurons with calcium overload, and the percentage continued to drop as a result of treatment. At the end of treatment, we saw nearly all of the neurons with calcium overload having recovered their calcium homeostasis compared to the control antibody. Therefore, aducanumab immunotherapy restored neurite calcium to control levels in TGD5 76 months. So conclusion of the first study was that treatment with aducanumab cleared amyloid plaques and restored neuronal calcium. So at that point, we went into the second study to determine whether what the aducanumab mechanism of action is. So the question we wanted to ask was, what type of A beta is aducanumab targeting? So in this case, we divided all of the A beta into 3 categories: monomers, oligomers, and plaques. And we asked whether aducanumab targets these different categories. So let's start with plaques. I just showed you data from our work, and there was supporting work from a publication by Biogen showing that their antibody does indeed engage amyloid plaques. We had, in addition, the Biogen publication reported that aducanumab does not engage monomers, hence some of that synaptic activity effect of A beta that Chris mentioned earlier today remains intact as a result of aducanumab treatment. So the next question we want to ask, does aducanumab target oligomers? And the reason this question is important is because there's been converging line of evidence to suggest that it's the oligomers that are the toxic species in Alzheimer's disease patients' brain, and there is substantial evidence from animal studies, culture studies in support of this. Also, if you can imagine, if immunotherapy is the approach to treat Alzheimer's disease, these large antibodies have very difficult time crossing the blood-brain barrier. So the small amount that actually crosses the blood-brain barrier has to target the toxic A-beta species. And if oligomers are those species, it would be beneficial to target those with the antibody. So that was the question that we set out to answer. And the oligomeric A-beta preparation that we chose to study are adults, which are amyloid beta derived diffusible ligands. It is a synthetic form of amyloid beta 42. Why did we decide to go with the adults? As you know, there are many different oligomeric A-beta preparations. Well, according to Bill Klein and colleagues and his adult hypothesis, which as you can see looks very similar to amyloid hypothesis, elevated A-beta 42 levels due to increased production or decreased clearance result in A-beta 42 assembly into adults. Adults bind with high affinity to neuronal receptors and trigger aberrant synaptic signaling. which results in failed information storage, aka LTP blockade, memory impairment, and dementia, through the mechanism of persistent downstream signaling, synaptic degeneration, and cytoskeletal collapse due to neuronal death. Adults are present in APP mouse brains, as you can see here. They're present in the hippocampus as well as the cortex. Our recent work has shown that adult application leads to calcium overload in vitro. Here we're looking at primary neuronal cultures with a calcium reporter in the one. At baseline, the neuronal calcium levels are normal, within the normal range. However, after direct application of adults to these neuronal cultures, you can see that there is calcium elevations. In addition, adult application leads to calcium overload in vivo. Our work has also shown that topical application of adults to wild-type mouse brains results in elevation of calcium within cell soma, as well as in neuronal processes. And adult application directly to the mouse brains, healthy wild-type mouse brains, leads to disruption of circuit activity. Here we're looking at the slow oscillations that are disrupted after adult applications compared to the wild-type animals. In addition, Bill Klein's work has shown that adults kill hippocampal neurons in slice cultures in addition to dissociated neurons. As you can see here, the percentage of dead cells increases with increasing concentrations of adults. And adults block LTP compared to the control. Adult applications significantly inhibits LTP. So then we wanted to see whether aducanumab would target adults and neutralize the toxic effects of adults. So we performed immunodepletion study where we took aducanumab, we took adults, we combined them together, and then we used magnetic beads to pull out the aducanumab adult complex and dissociated that complex, and then we measured amyloid beta in the supernatant that was left over after the complex was pulled out, and also we measured how much A beta was actually pulled out in the elute complex. So compared to no antibody treatment, we saw a substantial reduction in adult concentration as a result of aducanumab treatment. So aducanumab does engage adults, and it is able to pull it out in our immunodepletion experiments. Interestingly, 3D6 also was able to decrease the adult levels, the A beta 42 levels in our supernatant, and pulled it out in the elute. So what we did next, we took our supernatants, or the adults that were immunodepleted with the antibodies, and we've applied these supernatants directly to the primary neuronal cultures. We also recorded, monitored calcium concentrations within these neuronal cultures. As you can see at baseline, the neurons are maintaining their calcium homeostasis. However, after application of adult, there's some cells that can be found that have calcium elevations, shown here in red. When we apply reverse adult that has no effect, we saw no calcium elevations within those neurons. So what happened when we applied adults that were immunodepleted with antibodies? When we applied aducanumab immunodepleted adults to neuronal cultures, we did not see as many neurons with calcium overload, or these high levels of calcium, suggesting that aducanumab, it was neuroprotective. In addition, in this 3D6 condition, we didn't see that many cells with calcium overload. However, when we treated adults with the control antibody, that antibody that does not recognize A beta, we saw elevations in neuronal calcium within our neuronal cultures. Therefore, immunodepletion with aducanumab prevents calcium overload after application of adults to primary cortical cultures in vitro. And this is the quantification. Here, the percentage of neurons with calcium overload in baseline was very low. However, that percentage increased after application of ADL. Reversed ADL had no effect, and application of adults that were immunodepleted with aducanumab failed to significantly increase the percentage of neurons with calcium overload, as did immunodepletion with 3D6 antibody. Immunodepletion with the control antibody, however, that supernatant did elicit calcium overload in our primary neuronal cultures. So aducanumab targets oligomeric A beta as well as fibrillar amyloid plaques. So then the story continues. In March of 2019, Biogen and Eisai reported that they halt phase 3 aducanumab trials due to futility analysis, and that was really disappointing for the field. And then in October of 2019, they reported that, well, actually, we take that back. We might have seen an effect. So let's go through the steps a little bit. And Biogen reported in their investor relations website, tried to explain what really happened. So the futility analysis was based on earlier and smaller data set. There were 2 clinical trials conducted, Emerge and Engaged, which were both discontinued due to futility analysis. After discontinuation, additional data became available, and Biogen consulted with external advisors and FDA. They concluded that Emerge was actually positive. There was an effect, while Engaged believed to be supportive. And they plan to file to FDA at that point. Here are the doses that they administered. This is the dosing regimen. Some Alzheimer's. So this is really important. They enrolled patients with mild cognitive impairment due to Alzheimer's disease and mild Alzheimer's disease dementia. So these are very, very early stages of Alzheimer's disease. They administered either placebo, a low dose, or a high dose of aducanumab. Initially, ApoE4 carriers were to receive either the low dose 3 mg per kg or 6 mg per kg. However, there was a protocol amendment that sent these 6 mg per kg patients onto the 10 mg per kg dose. And that is what they explained that really made a difference in their result. This is the time course of what actually happened. These were very long clinical trial studies. There were several patient populations. The futility analysis was conducted on opportunity to complete population. However, the larger data set that was analyzed was opportunity to complete as well as intent to treat. So the futility of analysis was conducted, if you can see here, the number of subjects, close to 50% for Emerge and a little over 50% for Engage. However, in the intent to treat, there were substantially higher number of patients that were analyzed, and a fraction of those received amyloid beta PET scans in both Emerge and Engage clinical trials. And once the larger populations were analyzed, the data from those patients was analyzed in the high dose. And again, remember that there were those patients that were, due to protocol amendment, that were sent to 10 mg per kg, the higher dose, and inclusion of those patients is what really made the difference. So the patients on high dose of aducanumab had a statistically significant slowing of cognitive decline when measured by this cognitive test in both the intent to treat population and the other population. The low dose, however, was not statistically significant. In addition to slowing of cognitive decline, Alzheimer's patients saw a dose-dependent decrease in amyloid PET. So here's the data. This is the time course of placebo, low dose, and high dose in eMERGE clinical trial, and this is placebo, low dose, and high dose in ENGAGE. And as you can see here, there's a dose-dependent and substantial decrease in amyloid that was seen on PET scans of these patients, especially in the high dose. So this was really exciting and optimistic result. And Biogen completed submission of biologics license application to FDA for aducanumab as a treatment for Alzheimer's disease. And they enthusiastically mentioned on their website that if approved, aducanumab would be the first treatment with the potential to meaningfully change the course of Alzheimer's disease. With that, I would like to end my presentation. I would like to acknowledge the present and past members in my laboratory. Special thanks to Alyssa Russ, who helped organize this symposium. And my mentors, our collaborators, of course, our funding sources for all of the work that is funded. And thank you very much for your attention. Now, last little bit, I do have a conflict of interest. The studies discussed here, both of the studies were funded by Biogen. And with that, I will take any questions that you might have.
All right, we can open the floor to questions now. There are a bunch in the chat, so